Welcome back, everyone. So we were just talking about our membrane lipids, or the fluid part of the cell membrane. But the fluid mosaic model, we need to get into the mosaic part, and that is our proteins. So this does include some lipid domains when we talk about the mosaic, but the proteins are really the big players in this mosaic that we're going to talk about. A lot of how we've studied the proteins and the fluid mosaic model came from something called freeze fracturing. So you freeze a cell membrane, and then you hit it with the diamond knife, and it chunks it open between your two different layers, your extracellular layer, or the part of the bilayer that's against the extracellular fluid, and the cytoplasmic layer, or the side of your membrane bilayer that was facing the cytoplasm. And you can open it up and see what is inside of this lipid bilayer. When we've studied it, we can find different types of proteins here. There are membrane proteins that have different types of hydrophobicities, and because of that, they're going to have different positions in the membrane or on the side of the membrane. And the way that they are intercalated into that membrane is going to have different impacts on how easily they can be extracted from the membrane. And we end up with three kind of general categories for membranes. There are integral proteins. They have been integrated into that phospholipid. It might be integrated just a little. It might be integrated where it goes all the way through, or it might be integrated where it goes through multiple times, or multiple pieces attached. Those are all integral proteins. Then there are peripheral proteins. Peripheral proteins are on the periphery of the plasma membrane. So here you can see it's not going into the membrane, but it's still attached to it. And then there are lipid-anchored. So here's a protein, and it has this little lipid tail that is anchored into the plasma membrane, even though the rest of this protein is outside. So that little lipid part is going to be in here. This is an isoprenyl anchor. This is a GPI anchor. Either way, it's attached to that cell membrane because of this little lipid tail it has. Integral membrane proteins are embedded in the lipid bilayer because they have hydrophobic regions. This is a hydrophobic region, this is a hydrophobic, whatever part is going through the plasma membrane is a hydrophobic region. Peripheral proteins do not have hydrophobic regions. Peripheral proteins are hydrophilic, so they're going to be maintained within the cytoplasm or extracellularly, just on the surface of the bilayer. Lipid-anchored proteins are hydrophilic, so it is still on the inside or outer layer of the plasma membrane, but they have covalently bonded lipid molecules, and those covalently bonded lipid molecules anchor it into the bilayer. Most membrane proteins have one or more hydrophobic regions. Those hydrophobic regions are going to want to interact with the hydrophobic regions of the cell membrane. So integral membrane proteins have their hydrophobic regions embedded into the interior of the membrane, and they're going to be more difficult to remove. So these proteins can be dissolved a little bit more easily, but these are going to be interacting with that plasma membrane, and it's going to be really hard to get them out. Some integral proteins are called integral monotopic. So mono meaning one. They are embedded in just one side of the plasma membrane. But most membrane proteins are transmembrane. That means they go across the membrane. So they are going to have an intracellular side and an extracellular side. Transmembrane proteins can cross the membrane just once. That would be the single pass protein. Or they can pass multiple times. So this protein starts inside, then it goes out, then in, then out, then in, then out. So it's got multiple hydrophilic regions and multiple hydrophobic regions. The transmembrane segments of transmembrane proteins are going to be hydrophobic. That hydrophobic part that is embedded in the lipid bilayer is the hydrophobic region of the protein. Most of the time, those hydrophobic regions are an alpha helical configuration. They're usually about 20 to 30 amino acids long, and those amino acids will have hydrophobic R groups. Although they don't have to be this alpha helix, that is the prominent domain we see, but sometimes they could form a beta barrel, which is a modification of a beta sheet that kind of forms more of a barrel shape. You typically see a beta barrel sheet more often in something that is forming a pore, because it can kind of wrap around to form that barrel, like a barrel running through the interior of your cell membrane. Single pass membrane proteins will have the C terminus of the protein extending on one side of the surface and the N terminus on the other. Although it doesn't have to be this side intracellular and that side extracellular. For this protein, it'll have this arrangement. For another protein, it might be C terminus outside, N terminus inside. So for each protein, they're going to have a specific, this is supposed to be outside and this is supposed to be inside. But that doesn't mean that it is going to be the same way for every single protein. Glycophorin would be one example of a single pass membrane protein. Glycophorin is a protein found on the erythrocyte plasma membrane. The way that it is arranged is so that it has its C terminus in the inner surface and its N terminus on the outer surface. And you can see on the N terminus we have lots of these carbohydrates, which are going to play important roles in the identification of that cell. Multi-pass proteins will have 2 or more, even up to 20 or more transmembrane segments. One that we know a lot about is bacteriorhodopsin. Bacteriorhodopsin has been studied a lot. It is a membrane protein found in archaea. It actually is purple in colors than purple. So this is right on the dot to color in purple in the picture. So bacteriorhodopsin has 7 transmembrane portions, and it forms a channel. This 7 transmembrane proteins is a very common structure that we see in a lot of our channels, but they cross the membrane 7 times. So 1, 2, 3, 4, 5, 6, 7. That means that it has its C terminus inside and its N terminus outside. Peripheral proteins are going to be proteins that do not have a discrete hydrophobic region. It might have a few hydrophobic amino acids in it, but not enough to give it an actual hydrophobic region of it. So they are not going to penetrate the lipid bilayer. They are just bound to this inner surface or outer surface of the membrane through weak electrostatic forces and hydrogen bonds. They might have a few hydrophobic residues that help anchor them to the membrane surface, but they can generally be removed by changing the pH or changing the ionic strength, breaking those hydrogen bonds, and purifying out that peripheral protein. And then lipid-anchored membrane proteins have polypeptide chains, and they're going to be on the surface of the membranes. And they're covalently bonded to lipid molecules. Those lipid molecules then can embed into the bilayer. Proteins bound to the inner surface of the plasma membrane are linked to fatty acids or isoprenyl groups. You can solubilize the membrane proteins and extract them from cell membranes, and then they're able to be studied. Usually this is easiest to do with peripheral membrane proteins. You just have to change the pH or the ionic strength, so how much saltiness is there. Chelating agents or cation binding agents can also be used to solubilize peripheral membrane proteins. Lipid-anchored proteins can be isolated by similar means. It's a little bit trickier, a little bit harder to isolate those transmembrane spanning proteins. To isolate an integral membrane protein, you're usually going to need to use a detergent. A detergent is something that will disrupt that lipid bilayer. So you have something like, here's your lipid bilayer, here's your transmembrane protein, here's your detergent, and it just forms this little micelle, this little detergent bubble around your protein so that it is disrupted, or so that it is pulled out of that membrane. This also means when it's pulled out of the membrane, its function is probably going to be distorted. Its shape may be distorted a little bit. So this isn't a great way if you're wanting to study actual biological function within the cell membrane, but you're able to purify that protein to figure out what it has in it. Probably get a pretty good idea of how it's going to take its three-dimensional structure, and maybe even start to be able to figure out what it's doing. When you solubilize a membrane protein, you can do something called gel electrophoresis. This is going to separate proteins by size and charge, so you end up with smaller molecules that fit through that gel a lot faster, larger molecules that fit through it, they move much slower through it, so they stay at the top. So you end up with your proteins being separated by size. Then you have these little blue things that are supposed to be antibodies. The antibodies are going to bind to the specific protein. So if you're looking for bacteria rhodopsin, you use an antibody for bacteria rhodopsin. You take an antibody specific to the protein or the polypeptide you're looking for, and it's going to bind to your protein layer. And then you can take a picture of it, and your antibodies are going to fluoresce and give you a line here. So you'll say, oh, that's where my protein is found. I know how big it is then. I can tell how much it moved. So that is Western blotting in a nutshell. That's how you do Western blotting. And that's important to be able to identify which proteins are in a sample and the relative abundance. If you have a bigger band there, you know you have more. If you have a smaller band, you have less. If you don't have any showing up, that means you don't have that protein at all because you don't have any antibodies being bound. X-ray crystallography is another way that we can determine the three-dimensional structure of proteins. You may remember X-ray crystallography from talking about learning the structure of DNA with Rosalind Franklin. This is how she did her experiments. But anything that you can crystallize, you can figure out its structure with X-ray crystallography, including proteins. Integral proteins are usually difficult to isolate and crystallize. We've developed better techniques recently, so they are starting to be studied. And over 900 membrane proteins have been studied this way. You form a crystal, you shine an X-ray through it, and that crystal will diffuse those X-ray particles, and you come up with a sort of shadow band picture. And this is not something that I would be able to deduce myself, but that then tells you what that three-dimensional structure of that protein is. You can also do something called a hydropathy analysis. So if you know the protein sequence, you can stop and analyze each R group on each amino acid. How hydrophobic or hydrophilic is it? And then you make this hydrophobicity plot. So here, this amino acid is more hydrophilic, this one's a little bit hydrophobic, and you just come up and measure each amino acid, where it would fit on this. And from that, you get this plot that shows you there are these 4 spots that are very hydrophobic, and there are these regions that are more hydrophilic. So you can figure out this is going to be in the cytoplasm, this is in the cell membrane, this is outside the cell, in the cell membrane, inside the cell, in the cell membrane, outside the cell, in the cell membrane, inside the cell. Just looking at these regions that are hydrophobic or hydrophilic. So you can come up with and determine that your cell structure has some sort of structure like this. It is a tetraspan, and it spans 4 times through the membrane. These are also important types of proteins that we'll see. You can use a computer program to figure out how hydrophobic or hydrophilic each of your amino acids are to get this hydropathy index and figure out these different windows for what is in the cell, what is within the cell membrane, and what is on the interior or exterior in the hydrophilic regions of the cell or its environment. Okay, we just have a little bit more to study about our protein function within our cell membranes. So we are almost done with this chapter. I'll see you back in a little bit. Bye.